Welcome everybody. It is my greatest pleasure to be here today and to be able to present you our work on the effect of infrared light on protein interfacial water and consequences for protein-protein and protein surface interactions. So I'll start from saying that water is inherently bound to protein functioning. For example, it has been shown that for protein to perform the motions, it is necessary for water of hydration to perform translational motion. Then, for example, Professor Pollack has shown that on muscle contraction, there is a loss of water structuring and then restoration of the structure in a resting state. Then there is water-driven molecular recognition, for example, between the protein and its ligand, in which the hydrogen-bonded water molecules act as an adhesive glue, linking the protein and a ligand, starting from the early intermediates to the final self-assembled complex. Then, for example, water molecules can form those hydrogen-bonded water wires that will conduct proton, and they will confer the conductive properties to the protein interfaces. And all those and many other observations led to the conclusion that protein, together with its hydration water, should be treated as a single biologically active entity. And hydration water is not restricted only to the 1 or 2 layers of water molecules associated with the protein surface, but it can reach, for example, 10 layers of water molecules as shown by the tetrahertz spectroscopy studies. And water molecules in this hydration shell, they are highly correlated in terms of their dynamics and their structure. So they have some collective properties. For example, they have the structure resembling the liquid crystalline structure, and they have the net dipole moment. So we have the dipoles that are composed by many aligned water molecules. So because of these important functions of water, if we have the way to affect the hydration of protein, it is also the way to tune biological or bio-inspired processes. And if we can recognize how to affect the hydration, what affects the hydration, we can better understand how the living matter works. So I was really, really greatly inspired to find out that we in fact have a tool, a remote physical trigger that can affect protein hydration. And this inspiration comes from the works of the group of Professor Pollack. And this works, as everybody here knows, showed first of all that water near hydrophilic interfaces is structured, and it is structured to the extent that it excludes many salts, so it was called exclusion zone. And what builds the structured waters is the incident radiant energy. So here is the image showing the increase in the volume of structured water near the hydrophilic interface in response to the radiant energy. And the most effective in building the structured water was the infrared light. So it works for hydrated hydrophilic surfaces. It should work also for protein. So we decided to check if we can in fact see that infrared light can induce increased structuring of the protein interfacial water. So in order to do so, we performed simple experiments. We prepared the aqueous solution of the proteins and we exposed them to the infrared light emitted by the light emitting diode for the 10 minutes. With respect to the characteristic of light, we used 2 LEDs with the emission maxima at 2.9 micrometers and 3.2 micrometers. And these are wavelengths that are very strongly absorbed by the liquid water. This is the infrared spectra of water. And then we measured the infrared spectra of our irradiated protein samples. And this is the result. So this is the infrared spectra of the aqueous solution of lysozyme at different temperatures, at room temperature, but exposed to the infrared light. And this part of the spectra reflects the OH vibration on formation of hydrogen bonds in water. And in general, you can see that the effect of infrared light is comparable to the decrease in the temperature. So 0 means, in fact, protein solution stored in ice. So what happens when we decrease temperature? Of course, we increase the number and the strength of hydrogen bonds in water, so we increase the water structuring. So it seems that, in fact, infrared light has the same effect, so it increases the structuring of water near protein interfaces. from each of these spectra, the background water spectrum was subtracted. So this only comes from the interfacial water. And this is kind of interesting observation because everybody knows that infrared light is a source of heat, and its effect is comparable to cooling of our solution. And you have one more very interesting comparison. And this is again the spectra of the aqueous lysozyme. This time this is the lysozyme in an unbuffered aqueous solution. A lysozyme placed in a buffer at pH 4.5, so this is far away from the protein isoelectric point. And again, spectra of lysozyme in unbuffered solution but exposed to the infrared light. So this time you can see that the effect of infrared light can be also compared to the effect of placing our protein in a buffer. So what our buffer do? It's far away from the isoelectric point, so the protein is highly charged. If it's charged, of course, the charges interact with water dipoles, they will structure the interfacial water. Yeah, so it again kind of confirms that the infrared light has in fact this water structuring ability. And talking about the charging of protein interface, there is one again interesting result. If we in fact measure the zeta potential of our proteins exposed to infrared light, we can see that it increases in magnitude in response to infrared light. So 0 means the control samples, so non-irradiated samples, and this is the increasing intensity of light. So for myoglobin, you can see that irradiation makes the zeta potential more negative, and for lysozyme, when it's positively charged, the irradiation makes the zeta potential more positive. So it seems that infrared light in fact charges the protein interface. So the question is, why should it be the case? So if we accept that the infrared light induces the structuring of this protein interfacial water, and we have here some charges accumulated at the protein surface, then if we increase the volume of this hydration layer, we can accumulate here more counterions, and we can also make this water more polarized. So the accumulation of counterions will be enhanced. And this is what we measure as zeta potential. We measure these mobile ions at the boundary of this hydration and the bulk water. So up to now, our investigations kind of proved that infrared light has in fact this structuring effect on the protein interfacial water, and that it can effectively induce charging of protein interface. And we knew also from the studies of Professor Polak that for this charging to occur, we do not necessarily need the counterions in the solution, because the water itself, the structured water, can separate charges. And in fact, in my work, the proteins were put only in water without any background electrolyte. So knowing this, we wanted now to check if we can see apart from seeing the effect of infrared light, if we can see manifestation of this of its influence on some protein-based interactions. And here again, we had already some clue from the colloidal science. In which, if we consider our proteins in great approximation as some macro ions of the same charge, and then because of those counterions present in the boundary of this hydration layer and the bulk, we can have attractive interactions between those macroions of the same charge because of the interaction between surface charges and the counterions. So they can come together, they can associate. And in fact, a group of Professor Polak also showed that if we irradiate by infrared light the colloidal particles, we can induce their attraction. So we wanted to check if it works for protein as well. And I was working in the field of protein crystallization. So again, we prepared the same aqueous solutions of proteins, but this time we added here also precipitants, usually inorganic salts, to force our protein to come out of the solution. We irradiated with the light of the same characteristics, and we switched off the light and waited for crystals to grow. And these are our results. So in the control conditions, we, by purpose, diminished the concentration of the protein so that we could not induce any crystallization at all. And if we exposed proteins in such conditions to infrared light, we could grow beautiful crystals of hemoglobin, nice crystals of myoglobin, and crystals of DGAOR. And important for crystallographers, these were all diffracting crystals. So the answer why the infrared light should have such nucleation-inducing potential can come from what I have shown just a while before. So from those attractive interactions coming from the separation of charges in protein interface. There can be also one more contribution, namely it is recognized that the main driving force for protein-protein contact formation and crystallization is the release of the structured interfacial water. So if we induce more structure here, we have more entropy gain on releasing the water when the crystal contacts are formed. So it can also contribute to this nucleating effect of the infrared light. But apart from this, we made one more very interesting observation. We again, by purpose, changed the conditions in our crystallization drops. This is for hemoglobin. That at control conditions, we had jelly precipitate formation. And it generally means that interactions went too fast. There was no time for proteins to find their proper geometrical arrangements. So this is non-specific aggregation. And if when we exposed proteins at those conditions to the infrared light, we could form three-dimensional crystals. So we avoided this non-specific aggregation. And this is kind of important because many neurodegenerative diseases are associated with the non-specific aggregation of proteins. So we suggest that that structured water, apart from being able to induce those attractive interactions between biomacromolecules of the same net charge, it also acts as a protective layer for proteins. So they can freely rotate and they can find the specific orientations between the contacts before the contacts between them can be formed. So up to now we are able to show that in fact infrared light induces structuring of protein interfacial water, that because of this structuring we have some charge separation in the protein interface, that this separation of charges can induce attractive interaction between protein, and that structured water acts as a protective layer for proteins. And now we moved in our investigation to the other kind of protein-based interactions, namely protein surface interactions. And for those studies, we selected different proteins of different size and isoelectric points. And as a surface, we selected silica nanoparticles. Those interactions are important because for the nanoparticles, in fact, their interactions with the environment depend on their surface characteristics. Of if there are proteins adsorbed at the surface, for example, what the cells see will depend on the characteristic of this so-called protein corona. And also, effect of those adsorbed protein on the particle-particle interactions are also very important because the ability of nanoparticles to cross different barriers, like entering the cells, depends on their size. So if they aggregate, they may not be able to cross the barriers. So in order to study those interactions, we used the sediment volume method. We again prepared protein solutions in water, in salt, and in a buffer. We irradiated them with the infrared light for 10 minutes as before. And then we transferred those irradiated solutions to the dry silica nanopowder. We gently mixed the solutions and allowed them to sediment. And now, then we measure the sediment volume. So in this simple approach, the more hydrophobic the particles will be, the more compact sediment they will form in solution. So this sediment volume can be correlated with the hydrophilicity of the constituent nanoparticles. So we can move to the results. These are first results without the infrared light. So if you look at the results for the proteins in water, it can easily be concluded that the sediment volume increases with increasing hydrophilicity of the protein coating our nanoparticles. So the lysosome is the most hydrophilic. Now albumins are the larger proteins that are more hydrophobic. And this hydrophobicity is measured by different probes and is also expressed in the conductivity of those proteins, because the smallest proteins with the higher charge density is the most conductive one, of course, and is the most hydrophilic because it has the highest amount of charges that interact with water dipoles. And those that are larger and has lower charge density, they are also less conductive. And now we should also to understand further results, look what happens when we change our environment from the water to salt. So here we can see two different types of behavior. So for lysosome, the sediment volume decreases on placing our system in salt. Now for albumins, in fact, the hydrophilicity of the sediment increases on changing the environment to the salt. So to understand this, we have to consider what happens when we are adding salt to our protein. So this is a very simplistic model of the protein. When the violet area is the hydrophobic area, the green balls are the charge residues. So if we add the salt, first of all, we have charge screening effect, of course. So if we screen the charges here, we diminish their potential to interact with water dipoles. So we diminish the hydrophilicity of our of our proteins, and this is the effect that we observe for lysosome. So this is first of all, this is the charge screening effect, and this also is kind of corroborated by the observation by the group of Professor Pollack that adding high concentration of salts diminishes the structured water. Now, how can we explain the increase in hydrophilicity in case of albumins? So albumins, they are larger proteins. They have larger solvent-exposed hydrophobic surface area. And apart from those charge screening effect, we have here also adsorption of the ions, of the weakly hydrated ions, to the non-polar protein surfaces. And these adsorbed ions, because they remain partially hydrated in water, they will confer additional hydrophilicity to the proteins. And this effect prevails in this protein, so that's why adding salt for albumins in fact increases the hydrophilicity, which is expressed also increased sediment volume. So remembering about those interactions, we can now move to the effect of the infrared light on our sediment volume. So this is already change in the sediment volume in response to infrared. The positive change means increase in the sediment volume, so increased nanoparticles hydrophilicity. Now, negative changes means decreased sediment volume, so reduced particle hydrophilicity. And this is how our system looked like in reality. So you can see that the changes in the sediment volume are microscopically visible. So now, if you want to explain the increase in the sediment volume, increase in hydrophilicity, like observed for lysozyme and for Bowen serum albumin in water and salt, then we can recall the theory that we were talking about today. Sorry, it doesn't want to work. That infrared light induces the interfacial water structuring. So if it induces this structuring, it increases the hydrophilicity of our protein, it increases hydrophilicity of the solid. And this is also expressed in our results from the quartz crystal microbalance. So here is the frequency change, which is proportional to the adsorbed protein mass when we deposit here lysosome on the silica sensor. And this is dissipation change, which is indicative of the system softness. So this is for a non-irradiated system. And look what happens when we deposit the irradiated protein on our silica sensor. So the difference is quite substantial. Here we have the very characteristic spikes in the frequency and in the dissipation. And such features are interpreted in the literature as the indication of the adsorption of the highly hydrated molecules or molecular assemblies that then spread on the surface, and then they lose the water of hydration. So the system becomes less soft, and we lose some mass because of the loss of water. So it in fact confirms that this irradiated lysosome, it is more hydrated, and then it loses this water of hydration when it has to accommodate at the surface. So how can we explain now that infrared light decreases hydrophilicity for albumins? So in fact decreases the sediment volume here. To understand this, we have to recall what happens in those observed for mainly in salt and in buffer solution. So to understand this, we have to recall what happens with those proteins when we add some ion to the solution. So we have this adsorption of the ions to the non-polar surfaces, non-polar protein surfaces, and this was the effect that was conferring the additional hydrophilicity to the proteins. Now, if we irradiate such protein, we increase the hydration of those charged interfaces, and also the charged interface created by those adsorbed ions. So, if those ions become more hydrated, it seems that they may eventually have more propensity to bulk water than to the protein surface, so they may desorb from the surface and leave our protein more hydrophobic. What would be expressed? What would be, and this effect would be expression of this increased hydrophobicity. And this increased hydrophobicity is also confirmed by. Indirectly confirmed by our result from quartz crystal microbalance. This is Bowen serum albumin adsorbed on the silica sensor. For the irradiated system, we have here much more mass adsorbed on the surface than for the non-irradiated system. And it would be in agreement with the fact that this more hydrophobic protein has more propensity to separate from the bulk solution and to adsorb on the surface. So if we consider our proteins that are brought into close contact, like on nanoparticles in a sediment, then their tendency to aggregate will increase with increasing hydrophobicity. But we should also look what happens if we consider their interaction in solution. So here the situation is different. This is the size of the protein-coated nanoparticles measured in water. So this is the size of the bare nanoparticles without protein coating. When they are placed in water, they form large aggregates, about 300 nanometers. Now, lysosome-coated nanoparticles also form very large aggregates. Bowen serum albumin-coated nanoparticles form large aggregates, but for ovalbumin, we see that the size of those nanoparticles corresponds to the size of individual or eventually to the dimer of the particles. So why the ovalbumin should have such different effect on stabilizing of the colloidal suspension of our nanoparticles? So we can start from the other end. So to understand why those protein-coated nanoparticles can aggregate in solution, we can recall that for attraction between those like charged macroions, which that our nanoparticles will be, we have to have this interfacial charge separation, and that this charge separation can be provided by structured water. So it seems that in fact this lysosome that induces water structuring provides this charge separation in the surface. The same should be the case for Bowen serum albumin, and it seems that ovalbumin does not have this water structuring ability. And this is in fact confirmed by our observation from the quartz crystal microbalance. If you remember, these spikes in these neutral sparks for lysosome were indication of the highly hydrated molecule adsorbing on the surface, and here we do not have this effect for the albumin. And also if we look for the, I'm finishing. If we look for the sediment hydrophilicity, we see that already in water, in response to infrared light, we decrease hydrophilicity of our protein-coated nanoparticles. So it seems like there is in fact no water structuring ability for ovalbumin. So the answer at the molecular level, why is it like this, seems to lie in the presence of the basic functional groups of the protein interface. Basic functional groups, these are those generally that have those partial negative charges, so they like to acquire proton and become positively charged. And it's in the literature we can find out that the positively charged amino acid residues, they form stronger and longer-lived hydrogen bonds in water than negatively charged residues. And our lysosome is generally basic protein, so it has many of those basic functional groups and has very pronounced effect on the water structuring. And if we compare Bowen serum albumin and ovalbumin, and these are the mainly basic residues, so you can see that the amount in the ovalbumin is much lower than in Bowen serum albumin. So it seems that for those water structuring, we need on the protein interfaces, we need the presence of those basic functional groups. And on this, I would like to move on to conclusions. So we could show that the infrared light induces structuring of the protein interfacial water, that because of the structuring, we have some interfacial charge separation that can induce attractive interaction between protein molecules of the same net charge. And then our results also suggest that this infrared light can induce displacement of the ions adsorbed to the protein nonpolar surfaces. And both those effects, so enhanced hydration of the polar components and facilitated dehydration of apolar ones, can contribute to the proper protein folding and stability in aqueous solution. Because if the protein likes to interact with water, it's happy in water, but if it can dehydrate its apolar surfaces, it can properly form its hydrophobic core. And on this, I would like to thank you very much for your kind attention.
Many proteins in cells undergo post-translational modifications. They have phosphate residues added to them, acetyl residues, and so on, which change the way that they behave within the cells and change their interactions with other proteins. They have major effects on the behavior of cells. Have you looked at proteins to see whether post-translational modifications affect the behavior of the proteins in your system, and vice versa? Can exposure to infrared change the propensity of a protein to be modified?
I mean, this is something that I would love to investigate in the future, some more complex protein-based interactions, like namely the interactions with the ligands and things like this, because this is the very beginning. I guess it obviously has to work both ways. So the addition of something has to change how protein interact with water, and this affecting the protein hydration, for example, by means of light, can change how they interact with their ligands. Maybe sometimes we need the light, like to work, to function. But this is something that I would like to find out in the future.
Yes. In relation to the first part of the talk, have you tried to deconvolute the broad band of water to see what kind of water species you have in the hydration gel?
No, I didn't try. I just found in the literature that the band in this spectral direction, the increase in the intensity and the slight shift to the high wavelengths is associated with this increasing number cooperativeness of hydrogen bonds, but I'm not expert in the infrared spectra, so I would not be able.
Because we have done some similar experiments, but in lipid bilayers, and we can see different populations of water. So maybe you can try that in your protein. Another question, in the second part, do you think that the protein can be changed in the structure when it absorbs to the oxide or
to the surface,
to the particles?
I mean, the lysosome should, generally it was shown by many studies, should not change because it's a so-called hard protein, so there should not be too much structural change. And my nanoparticles are very small in comparison to proteins. There are 10 nanometers, and my proteins are like 2.5 to 7 nanometers. So unless they wrap around the surface, they should not change much, I guess. But there's, I didn't try to check specifically for it.
Thank
you. Here I am. So thank you very much for this very elegant approach. I appreciate it a lot. And this brings into my mind something that maybe you can add as an extra to our experimental setup, which is Gilbert Ling's approach on association inducting hypothesis, which means if you add ATP and potassium ions into your experimental setup, you probably can look between a denaturated protein structure and the one in the structural complex folded form, which should provide you extra evidence how much the hydration should look like and how much these changes between these two configurational manifestations. Mm-hmm. So would this be suitable for your experimental setup as an extra on for future investigations?
Mm-hmm. That's a very interesting point. Thank you.
I mean, we can talk about it later on anyway. Yeah.
Yeah. Yes. I was talking even about this, that this can be this thermodynamic contribuent, because it is found that in protein crystallization also, the main driving force is this entropy gain on losing the structured water. So if I'm structuring the water more on protein interfaces, there is more entropy gain if I'm losing this water on crystallization. So that's why it can also help on crystallization. So this was also, I consider this contribution, yes.